In this video, we will demonstrate how to analyze data from the Invitrogen QuantiGene Plex assay using the QuantiGene Plex Data Analysis App. This cloud-based software app is offered free of charge to all QuantiGene customers. All you have to do is sign up for an account at thermofisher.com and use the link shown. This diagram shows the overall workflow for QuantiGene Plex data analysis. A CSV file is exported from the Luminex instrument and then uploaded to the cloud. Once in the cloud, the user can define the plate map by identifying background wells and different treatment groups. Normalization genes are then selected and QC parameters can be reviewed. After running the analysis, the cloud software generates normalized expression data and fold change calculations. For more advanced analysis, chip files are exported from the cloud software and imported into the Applied Biosystems Transcriptome Analysis Console, otherwise known as TAC software. After logging into the cloud-based QuantiGene Plex Data Analysis app via the Thermo Fisher Connect platform, you will be directed to the dashboard where you can drag and drop the CSV file exported from the Luminex instrument. A green checkmark indicates that the data has been successfully uploaded. Opening the file in the cloud software takes you to the plate setup page where you can identify the background wells and different treatment groups by clicking on the pencil icon. The edit window will pop up and you can pick between unknown or background sample types. If you pick unknown, you are able to define or modify a group. This will allow you to group samples into different treatment groups or disease states as shown here in this edit window. The Add button lets you add groups. Click Save when done. After identifying treatment groups and background wells, you are going to select your normalization genes by clicking on the DNA icon in the top right navigation panel. Clicking on the DNA icon will bring up a list of the genes in your panel. You can then select which of those genes are the housekeepers or genes that you want to use for normalization by selecting the checkbox for the relevant targets or genes. If you select a minimum of 4 housekeeper genes, you can use the gene norm ranking function to rank the stability of each housekeeper, as it is important that the expression of housekeeper genes does not change between treatment groups. Any stability measure or M value less than 0.7 is considered stable. Click Save after selecting your preferred housekeepers or normalization genes. Quality control or QC parameters can be assessed by clicking on the magnifying glass icon in the top right navigation panel. There are 5 basic QC parameters under QC settings that can be customized: maximum background, saturation, limit of detection or LOD, minimum bead count, and technical precision as defined by %CV. Under View QC, there are pull-downs to view the data associated with each of the 5 QC parameters. As an example, LOD data is shown here with the LOD for each target in the panel displayed directly beneath the gene name. Failed LOD values are highlighted in red. There is also a function to determine assay linearity when clicking the magnifying glass icon in the top right navigation panel. For every new QuantiGene panel and/or sample type, it is important to run a set of serial dilutions to verify that you are within the linear range of detection for each target in the panel. You will also see the calculated % recovery here in a table. The Export button will allow you to export image files or tables. Click Return when done. The remaining icons on the plate setup screen include the hamburger on the top right, highlighted by the red arrow that allows you to reset the plate and restore replicates. There is also a help icon at the top of the screen that brings up the help manual for the software with detailed step-by-step instructions. When you are ready to run your analysis, click on the paper icon. Running the analysis will take you to the review screen that has a table of the normalized expression data for all of the targets and samples. The normalized expression is the signal of the target gene divided by the geometric mean of the housekeeper genes. Normalized expression values with an asterisk indicate targets that fail at least one of the 5 QC parameters. Normalized expression data can be exported as a CSV or Excel file using the export icon on the right. The download chip icon generates individual chip files for each sample, which can be imported into the TAC software for advanced analysis. Basic fold change comparisons can be calculated within this QuantiGene Plex data analysis software. A toggle on the review screen allows you to either set up comparisons between individual samples or between groups of samples. If you select a group comparison, then you will select a reference group, then one or more groups to determine fold change relative to that reference group. After setting up your fold change comparisons, you will get a list of fold change values and a fold change graph that looks something like this. You can select the scale of the graph at the top of the fold change list. Log 10 is generally recommended. If you hold the mouse over the bars in the graph, the fold change and p-values will be displayed. The list of fold changes and the fold change graph can be exported using the export icon on the right. More advanced analysis of QuantiGene data can be performed with the TAC software. The TAC software uses chip files which can be generated under the review screen using the download chip icon. The download chip icon generates a zip file that includes the individual chip files as well as the TAC parameter folder containing the library and annotation files. It is important to note that the zip file cannot be extracted with Windows built-in zip file support. The file needs to be extracted with third-party software that supports multiple archive types such as the open source application 7-zip or another similar application. TAC software can be downloaded via the link shown here. Before performing TAC analysis, a TAC library file must be imported and installed from the TAC parameter files folder that is exported from the QuantiGene Plex data analysis software. Click on the Preferences tab and use the Browse icon to locate the TAC parameter files folder. After assigning the TAC parameter files folder as the TAC library, the net affix username and password are automatically generated and the QuantiGene configuration file will appear as an installed array type. Each sample from your QuantiGene analysis will be exported as an individual chip file. These files can be directly dragged and dropped into the window under the New Analysis tab. You can also select the Import Chip File under the Import Files dropdown. Different experimental conditions or sample attributes can then be compared across samples during the differential expression analysis. Select Comparison from the drop-down menu in the header of the column you would like to compare. The title of column can be changed by right-clicking on the header. Type to assign condition values for each sample. Multiple samples can be selected and assign values simultaneously. Additional conditions can be added to the analysis by right-clicking in the header next to the previous condition or column by selecting Add New Column and Comparison in the drop-down. All conditions will be automatically compared in the analysis, but the Comparison Setup wizard can be used to customize the comparisons prior to running the analysis. After all of the comparisons have been defined, click Run Analysis. Gene lists and Venn diagrams can be generated under Summary View. Venn diagrams are convenient ways to visualize the intersection of genes from the lists generated by your comparisons. Scatter plots can be generated under Gene View to determine differential gene expression between two conditions selected in the drop-down. Specific data points lassoed in the graph are highlighted in the gene table on the left. Volcano plots can be generated to quickly identify statistically significant changes between two conditions by plotting significance on the y-axis versus fold change on the x-axis. Genes upregulated by more than twofold are shown in red, and genes downregulated by more than twofold are shown in green. Graphs can be exported as a PNG file by clicking on the hamburger icon on the right. TAC software can also generate heat maps using hierarchical clustering to group individual samples based on their gene expression. Finally, the TAC software can search the Wiki Pathways database for potential pathways associated with your QuantiGene panel. Relevant pathways are listed on the left and can be updated by clicking on the clock icon. Genes that are upregulated in your analysis are highlighted in yellow. Genes that are downregulated are shown in blue, and genes that are in your panel but are not differentially regulated are shown as grayed out. For more detailed information regarding the QuantiGene Plex data analysis, please refer to the QuantiGene Analysis Software Help Guide and the Quick Reference Guide entitled Use Transcriptome Analysis Console to Analyze QuantiGene Plex Data. Thank you for watching this video. For more information about the QuantiGene Plex assay, visit thermofisher.com/QuantiGene, and for more information about the QuantiGene Analysis App on Thermo Fisher Connect, visit thermofisher.com/QG Analysis App.